function as on-time data staging module into some scheduler. Now, this is the third part. Okay. And this framework is not complicated and works as follows. Very easy. So scheduler inquires whether data transfer is okay or not to data transfer scheduler when it attempts to map the job request to computer resources. If the data scheduler say, okay, a scheduler pin the start time of job and wait for the completion of data transfer. On the other hand, data transfer scheduler has a responsibility of monitor network throughput and manage network boundaries. So this is our environment. So our simple environment environment is is spanned over the US and Japan side. So right side is Northwestern University. In Northwestern University side, so the one of the VPN is located, and our side is a VPN also deployed, and also the prototype the schedule system is deployed on Osaka University side. So evaluation method is very easy. So we submitted assuming multiple users. We compare utilization of computer resources and waiting time with three methods shown in the next slide. When we submit the job, we change the ratio of jobs requiring the data transfer of data to the system. So we compare our method with two conventional methods. First one is the usual way. So user has to set up, deploy the data before submitting a job request. This is the usual way currently. And third one is okay. So if the job request is dispatched, when the job request is dispatched to the computer resources, then the data transfer happens. Okay. And second one, the middle one is our proposed method. So we compare the this slide shows the evaluation result, and so the data staging before job submission. So BSS is actually utilization is high, but waiting time increase with increase of the job ratio. And data staging after dispatch, so ADS way, this utilization decreased, but waiting time is short. On the other hand, so our proposed frameworks keep the utilization and irrespective job and waiting time is kept short. This is the research result so far. And from the next slide, so let me I go into the detail of the onion and main our planning in future. Yes. So again, this is the supercomputer system Squid. When we introduced supercomputer system named Squid, so we also built the data infrastructure named Onion. Onion's architecture is something like this. So the right part of the slide shows the Onion. So you know the supercomputer system is basically supercomputer system. Today's supercomputer system has deployed parallel file system for gaining the high performance reading and writing of the file. But in this time, so we try to deploy some kind of functionality of aggregating the data from the outside the system to supercomputer system. So you know that now AI people always told me that HPC people always think about computational resources first, computer system and the file system. But AI people say, okay, so AI people always think about data infrastructure, important. And after that, secondary, so computational resources should be on top of data infrastructure. That's why we set up this data aggregation infrastructure on trial basis. Okay, so Onion overview of Onion is like this. Okay, so next side is a parallel file system for. So this is basically this type of parallel file system is deployed to usual supercomputer system for today. And so so next to parallel file system, we set up the object storage for data preservation and data preservation and data preservation. And also we set up the interface that allows the scientists can to move the data with easily. Okay, this is this slide shows the internal architecture of our Onion data infrastructure. So basically, the S3 protocol is having taking a role of taking a central role. Okay, so by introducing S3 protocol and combine three different storage solutions, so now our user can move the data from the outside of supercomputer system to supercomputer system, and some user want to can make the computation result public right after computation. So now our user enjoying the liquidity and the portability of the data. Okay, and this slide shows actually this is not yet. This is our planning. So now our supercomputer system, supercomputer users are satisfied that the data can be moved easily. But one of the problem right now is the performance. Okay, so now, so our user say, okay, this Onion infrastructure is very good. So the infrastructure facilitates us moving the data. Okay, understand. But the performance, no. Okay. So actually, I already say, okay, this is the trial pages. So we have to increase the performance. And actually, now our university collects the money from the public people in Japan. And Osaka University had decided to invest the money to data infrastructure. This is our planning. Yes. So based on the success of using the DTN through the research, so I wrote this. So now in the campus, many research institutions and departments, there are many institutions which uses the enormous amount of data. And but one of the problem is that such researcher now bringing the hard disk for data sharing, even in the campus. So that's why we are now trying to set up the 400G or maybe 800G Ethernet to the campus and set up the DTN node, both of the computation center and research institutes. And now we are in the process of procurement. And so if we have succeeded, so now the research institution which have the scientific devices such as the microscope, so can move the terabyte plus data to the supercomputer system, and the user can enjoy the performance brought by supercomputer system. And right after the computation, such result can be shared to different different institutions. That is our plan. But this is that's plan. But maybe next APAN we can I can report the progress of status progress of this project. Okay, so this is summary. So sorry, actually I have my time is running out. So we actually enjoyed the DTN performance through the international collaboration, and we tested DTN experiment. Yeah, so I enjoyed. And based on the such success, we now I as a responsibility of supercomputing center now planning to and high-speed network in the campus. And maybe if we succeeded in deploying such scientific DMG in the campus. Next is to deploy such environment to the world globalized way. Okay, thanks so much.
Sloan schedule. Did you actually write that plugin yourself, or was it something that was so the Sloan component where you're holding the jobs waiting for the transfer? Is that a standard part of Sloan, or is it?
We have developed the pinning function. So that the function is なんだろう、pins starting time. That module is not standard part. So our student has to deploy. Yeah, I think we should do, but that is only currently prototype. So I guess we have to improve the quality. Maybe so after that we can. I forget.
And next presentation is on me. I prepared my slide. Okay, let me start. And thanks again is Dr. Susumi Pate-san. And he flight schedule is 2.5 in the afternoon o'clock. So he spent his the morning time to presentation in here. So let me start this my presentation. So my name is Jung-Hoon Moon, is from KIST in Korea. The KIST is the Korea Representative Supercomputing Center and Research Network Center. The name is the KoreaNet. Today I want to talking about is the APAN, the APRP working group status, and the Korea research platforms. It's kind of an overview, and also the include is the Asia Connect project from TNCC. And let me. Okay, and this is the contents of today. The first, let me tell you about this APAN, the work APRP working group. And second thing is about this KRP for domestic in Korea. And finally, I will talking about the new challenges and the related project in Korea. And from now on, I'll explain about the APRP in APAN. And this is the briefly the introduce APAN, the APRP working group. The APRP meaning is the Asia Pacific Research Platform. Since 2018, it has been initiated, and the goal is to share the XRP experience with the members and to propose the establishment of the APRP that will be part of the GRP. GRP meaning is a global research platform. The executive members are the chair and the co-chair and the secretary, and Andrew Howard, Asif Khan, and me. And so far, the APRP working group was total 10 times held working group meetings at APAN and the 20 sessions for last 5 years. The APRP working group scale is every 2 sessions and average 12 presentations from 8 countries of Asia regions. The average, the participant before COVID-19 offline is around 30 to 40, and the average participant during the COVID-19 online is around 50 and sometimes over 70. From 2023, the APRP working group we had as a hybrid. And our challenges, I wanted to introduce challenges. We got the grant, the Asia Connect project from TNCC, the name of building a high bandwidth distributed HPC. And I want to talking about the trend shift for so far until so far. When we start big data, we start as a big data highway. Big data superway is a kind of the required NNs or wait a minute. Yes. When we start is the big data superhighway that require the NNs upgrade in Asia. And then the shifted the building of the platform. The reason why the lack of IT infrastructure in Asia regions and also the distributed and the shared HPC-like environment needs. And then there is currently applying third-party applications such as weather, climate, and the environment, agriculture, and this kind of the smart farm, and AI, and the bio informatics, and the cloud, and so on. And the next about this overview of the Asia Connect, the project. The title is, as I mentioned before, is building a high bandwidth distributed HPC. The participant countries are Korea, Australia, Malaysia, and Pakistan. And duration is 1.5 years. The budget is around 150k euro. The and the co-PIs are the Andrew Howard from Australia, NCI, Asif Khan is Malaysia, from Peradana University, and the Noor Asila, Malaysia, from UPM, and Asif Plaza, and from Pakistan is Surkaibi University, and me. And this slide is a background of this project. Is as we know, the data explosion in science and technology drives the demand for high bandwidth, high capacity networks for accessing computers and storages and collaborations. At the same time, it brings many restrictions on the generation, transmission, and utilization of research data. The next two pictures are the very big motivated to this project. The figure one that shows the distributed computing resource integrated through the PRP infrastructure and the scientific result about analysis of CO2 using the distributed computing. It means that a specific the research drives the research results without the possessing high performance computing. The figure two that shows the urban flooding research using the distributed computing and the resources and the science data for LSTM, the analysis method for the climate analysis. And the infrastructure links the distributed platform and many career research institute through the National Research Network, which name is CREONET. The participated institute share the HPC, the resource for the researchers. The purpose of this project is to suggest the method to enable probably high performance computing resource to be delivered to underdeveloped and developing countries in Asia regions through TAIN network. As we know, there's most underdeveloped and developed countries in Asia is like IT infrastructure. So this is the basic concept for this project as shown in the picture. The step one is where science DMG-based high bandwidth networking architecture is designed in participating countries via TAIN. The step two, so we will interconnect available computing resource in a distributed manner. The step three, the resource of a computing service will be managed by the contained cloud technologies. It means it's the Kubernetes. To implement this, it is divided into three activities, and each will achieve the detailed goal. The activity one, the building of big data superhighway based on TAIN network, and the activity two, the building a distributed HPC platform. Activity three, the presenting the pilot user cases, and we selected the AI, science, and the bioscience. To building the big data superhighway based on TAIN is about this activity one. There are three steps enable this environment. The first one is interconnecting participant NNs via TAIN. There are CREONET, RNet, and the MyLAN, and the PRN. And the second thing is installing DTN, each participant institute. And the third thing is measuring the science teams in using the monitoring system. The more detail about the activity 1 would be presented by the Asipalaja and the session 2. And activity 2, to building the distributed HPC platform. And there are also 3 steps to enable this platform. And the building of the platform to CPU, GPU, computer resource, including storage. And the second thing is building blockchain-based ID federation technology. The third thing is building an easy user interface. Easy user interface, it provides an easy using for the students and the researchers. And they cannot handle this, the computing environment. More detail about the activity 2 would be presented in Kim at session 1, the next presenter. And this is activity 3, which is presenting pilot application user cases. These are the AI science and the bioscience for targeting ASEAN beneficiary in a high performance network environment, as shown the figures. It will be supported high throughput science research and contributed. All of the third party research topic stage now. The first one is in case of AI science, use cases by the Asif Plaza Sarkar University from Pakistan, the multiple object tracking for the aerial view imaging and the distributed training is kind of the MNIST, the running distributed MPI jobs. The more detail about the AI science of activity 3 would be presented by the Asif Plaza session 2. And the bioscience use cases by the Asif Khan from Perdana University in Malaysia, and applying high performance computing in drug discovery and molecular simulation, and the multiple sequence alignment MSA of a large number of sequences on HPC. More detail about this bioscience of activity 3 that would be presented by Asif Khan session 1. And also the other third parties research areas such as wireless and 6G would be introduced by the Q team at session 2. And the bioinformatics and the cost for automated goal would be introduced by the Professor Fitja from UPM and Alex Mora from the cost at session 1 and 2. So finally, just 5 minutes would be talking about this currently share the HPC status in Asia. As from now is talking about this KRP. This slide is to show us the big picture of a KRP deployment on the national scale. And that is established at the high reliability and high speed transfer system without boundaries between participants, and also support the research platform as your computing resources. Korea has 25 national research institute government funded. Most of them are located near KISTI, the same team as Daejeon. And so already Korea has a dedicated optical network for those institutes in the name of a Super Silent. So we are going to expand to major research institutes such as bio, aerospace, astronomy, earth science, and the chemistry, nuclear fusion research, and medical physics, ICT areas, and so on. So, and all the beginning steps, and then we will be gradually increased to cover all. And the strategy of these plans are the first one of step 1 is building the dedicated science, big data transfer, and the AI computing environment, which means that expanded the KRP. The step 2 is support the safe and the single domain transfer performance, which means that it is without any boundaries between them. And so step 3, the covering 20 national research institutes and the 4 universities of advanced science and technology. The expanding the national scale in Korea is in the viewpoint of technology, that is HPC over HPN. This slide is dedicated to support planned adding super facilities as a supercomputer data center and the GPU machines from KISTI. As you know, the KISTI is a representative of the regional, the supercomputing center of Korea, and also the operated Korean center. So we can drive the supercomputing and the data center, KRP, as a facilities of the via Creonet. It means that there's HPC over HPN, which is dedicated to the high performance computing in the data centric research areas. So from now, I will explain about this over the project and the proposal stage project-based platform. So we have three related research platform project. One is a smart hospital development project, and the other is a Korean Rural Development Administration project, and another is a Korea Environment Institute project. The first one is a smart hospital development project awarded September 2022. The title is the construction of the patient-tailored hyper-personalized digital medical ecosystem based on the Omniverse platform. The start of this project would be the April or May 2023. And the second one is a Korea Rural Development Administration project awarded this March 2022. The title is the development of an integrated linkage system for agricultural big data and the utilization model. And the participants are the Seoul National University and the KIST. We are going to develop some Kubernetes-based the container solution for the running model. The third is the smart decision-making system. The title is establishment and the development of a smart decision-making system based on diverse environment model. Our government wanted to the national disaster or the heavy rain or the earthquake, and they wanted to the right decision using of every data. So the funding by the Korea, the Environment Institute, and the participants are KEI, KIST, and the several related institutes, such as KMA and the Korea Rural Administration, and so on. And so they need this big data transfer, so they need analysis of the computing platform, and also including the GPU machines. As I mentioned before, all of our platforms can support these areas. This is the final station, is kind of the conclusion about the APRP working group APAN and the enhancement APAN, the APRP working group, the collaboration with the related working group, such as agriculture, the medical, and the disaster and the cloud in APAN. The finding is a new funding sources, such as after the Asia Connect project. The second one is about its deploy the research platform to Asia region and the Korea domestic part, enhancement of KRP to Asia areas through the TAME project, and the expanding to 25 national the research institute, and 4 AI and the high performance computing environment. The third one is about the new challenges. We want expanding to the disaster. It's kind of the urban flooding and the AI education areas. We want expanding to wireless areas, such like in the 5G and the 6G. And thank you for the listening. We have spent time in the early stages, 15 minutes. I want to save you time and just so quickly talking about that. Thank you. Anybody question about that? Okay. I want to introduce this. The next speaker is Kian Kim, and introducing of KRP and Korea platforms inside. Please. Thank you.
Yeah. Hello, everyone. My name is Kian Kim, and I'm working at KIST Korea. Yeah, I'm in charge of platform development in Korea Research Platform and Asia Connect project platform. In this presentation, I will introduce the Korea Research Platform and its current state of development and its future. There are various users and groups in Korea Creonet. Recently, various groups are requesting the need for platform and infrastructure for big data and AI. In fact, there are still institutions that exchange data using disk copy for big data transmission. In various groups, there are many organizations that configure and use infrastructure on a small scale to utilize computing resources. And the users talk about need for the various research environments for machine learning and AI research. To solve these problems, we are building and developing KRP. The purpose of the KRP is for free resource utilization and research collaboration among various research groups. It aims to efficiently share computing resources at the national level and help improve AI research productivity. KRP benchmarked NRP and PRP to build and develop a Korea platform. In the early days, the project called RNA Together was started as a small groups. KRP is a project to provide a system to users for the purpose of AI research, national research institutes, and science and technology universities. At the time, various discussions were held with a small number of national research institutions on network construction and AI research. For the same reasons as in the table above, requirements for an AI platform. Currently, a lot of development has been completed. So in recent cases, various projects are being carried out with various groups in Korea using data provided by the government. Most of the KIP platform was developed. Most of the KIP platform was built using open source. Open source platforms were installed for platform management, and the orchestrator was developed so that user can use them more easily. As open source is used to manage the platform Kubernetes, which creates and manages the most important containers of the platform. And the SAP storage are used for storage, and Harbor is used as a private image registry. For user login system, the ID federation system, CAFE developed by KIST is used, and CAFE login users are authenticated using keyclock. System monitoring used from Prometheus and Grafana, and the server logs and errors use Elasticsearch and Kibana. Open source is used by users to perform AI tasks or programming using R, MPI, PyTorch, and TensorFlow, Python, and so on. The interface basically provided by the platform is configured to utilize Jupyter Notebook. For all images, Jupyter Notebook becomes the base image. It also provides a command line interface, and reason why Jupyter Notebook is used as a base image is that the environment that links the authorization system will be linked to Jupyter Notebook in the future. The configuration diagram of KRP is shown in this figure. In our system, orchestrator and web interface have been developed for our functions to work organically. The functions provided by this platform are about 5 as follows. Consisting of login, computing, and storage, transfer, and monitoring. In the case of computing, it is divided into distributed computing and computing for individual users. In the case of distributed computing, AI work using GPUs of all distributed servers is possible. Or components provide the API and use this API to compose the web interface. In fact, the reason we provide the web UI is that there are not many researchers who can use it if the CLI version is provided to users. Therefore, the goal is to develop a user-friendly platform. Currently, in addition to developing the KRP platform, we are developing various big data and AI research platforms. One of them is the Asia Connect project platform. The Asia Connect project platform assignment platform is in fact being developed very similar to KRP. Development is still in progress, and master node and worker node have already sent and built equipment to Malaysia. This figure is a configuration diagram of equipment construction in 4 countries that are proceeding with Asia Connect tasks in the future. As shown in the picture above, 3 servers and 1 switch were completed in Malaysia last November. And the picture shown above is a picture taken after actual construction. And Australia donated 1 D10 server, and in the case of Pakistan, 1 switch and 1 server will be transferred and completed in March or April. Professor Ashku of Pakistan continues to help, and in Korea, Septridge is built and connected to Malaysia master node. Server construction for the Asia Connect project platform is almost completed when installed in Pakistan. And using the APRP.kr domain, we use the APRP.kr domain, and the platform open sources are installed, and the orchestrator and WebUI are configured. Currently, most open source is installation and the dashboard are configured. As shown in the figure above, it is already applied to configure the environment to access the dashboard. Only orchestrator configuration and the WebUI configuration remain. In the case of the WebUI developed is currently being delayed, so we aim to complete the configuration within April. We are working on 2 more projects. One is an agriculture, big data, and AI research platform development project, and the other one is environment, big data, and the AI research platform development project. The two platforms are being developed as very similar platforms, and the main purpose of to manage big data and perform AI. In fact, in the case of agriculture and environment, data from various models are linked to bring one figure. For example, to predict the strawberry harvest in this year, there must be various data values such as weather, insolation, sunlight, soil information, and precipitation at the time. Therefore, the focus of developing this platform is how to connect the models. Models developed for AI are composed of images, and these images are created as containers at the request of users. The created containers are linked using APIs. Also, it is intended to be constructed so that users can easily obtain results through mobile or web by of third-party applications. We have two representative use case using KRP. One is urban flooding. Urban flooding is a representative use case using KRP. As the weather changes rapidly in Korea, urban flooding is a big issue, and the Korea Environment Institute and the two universities have developed this AI model. And various experiments are being conducted on urban flooding. Last summer, 2 years ago summer, flood occurred in an apartment building in Daejeon, South Korea. At the time, it rained a lot in Daejeon, causing a lot of damages in the apartment. Daejeon City could not predict the flood damage to the apartment, and the flood barriers were installed later. Simulating urban flooding were performed, and then 3 flood barriers are installed around the apartment. If you look at if you look at the simulation picture, you can see that the water depths change with time. Before the water barriers is installed around the or around the apartment is red, and after the flood barriers is installed, it is blue. And the Korea Environmental Institute will continue to expand the scope with additional experiments. Second is agriculture environment. The figure below is a use case in which researchers in the agriculture field. It aims to improve the productivity of crops by performing various information that can make crop grow well through machine learning. The figure show explain of simulating the growth of crops using soil and weather. In this way, in the field of crops and the environment, convergence of various fields is required to have results to linking various models. Various institutions and universities are using KRP as follows, and we are trying to discover cases. Currently, requests are coming from university labs, and in the case of university labs, it is difficult to easily purchase expensive GPUs. Future research is continuous KRP system advancement and development because KRP does not currently provide distributed computing based on web UI. It is difficult for users to use it. In addition, we plan to find useful open sources for services and develop them into a better environment. We are continuously working and collaborating to increase the usability of the KRP. So we welcome both the project type and form that utilize the KRP. Based on the CreoNet infrastructure, we will continue to make effort to improve the productivity of AI research in Korea. Thank you. Thank you. Kirk. So I have one question. So several commercial cloud service is available. So did you include uh, your buying the commercial cloud service, the Google, Google GCP or AWS? Mhm. Yeah. Actually, we use the self-storage. Yeah, self-storage is use S3 protocol. Yeah, and as self-storage is use the object volume. Yeah, so we use the object interface. It is our default storage. Yeah, so we make a bucket. Yeah, so we use the bucket, and then bucket when bucket increase create the when we create the bucket at the time, do you know private key and the public key, and yeah, port, and yeah, and so on. Yeah, so and then we use the S3 protocol use, and then we platform transfer data and receive the data. Yeah. Yeah, protocol. We use just the S3 protocol. Yeah, and also one more thing. I told you, S3 protocol just used in server. So server and bucket and transfer the data. Also use the transport. Yeah.
Okay, thank you for your presentation. And the next presentation is the Professor Fijat from the UPM in the Malaysia. And he just online through the FUBA. Is please is on the screen her presentation.
Okay, okay, good. I think good morning in Nepal, and then here is already afternoon. All right. So for today, as a indirect user of HPC, I'm going to present or sharing a bit on of my research that actually we use the HPC in the fight against avian infectious disease. So this is the overview. So I will give a brief on the poultry industry in Malaysia and the disease and prevention and control of the diseases, and how actually the bioinformatics with the help of HPC could transform the poultry health monitoring, and then a bit on the research highlight and development, and also how bioinformatics help on leveraging the vaccine technology advancement, and conclusion, and also the way forwards. Okay, so basically poultry industry in Malaysia, has grown significantly at about 5% annually and has contributed about almost 90% of livestock production and 75% of the farm value of the livestock industry in the country. So basically the livestock is the second highest contributor to the value of gross output for the Malaysian agriculture sector. Is around 80.7 billion, where the poultry itself contribute about 16.8 billion. And Malaysia is self-sufficient for poultry meat and eggs, where Malaysia consumption is among the highest in the world, more than 50 kg per capita per year. So the industry is run commercially by large-scale integrators and also multinational corporation, and example of success story in the livestock industry in the country, and could perhaps act as a model for the livestock industry. So okay, the rapid growing of poultry industry, however, not free from many issues and challenges, such as high production costs and also emerging and re-emerging diseases. So for today, almost all non-major poultry diseases have been reported in Malaysia. So if you see from this one, this example of the pathogen that cause infection to the poultry industry, we have virus infection, for example, like NDV, IBDV, fall adenovirus, and also caused by bacteria, pathogen, fungus, and parasite like Salmonellosis, Pasteurella, Mycoplasma. So prevention and control of poultry diseases involve complex understanding of the interaction between the agent, host, and environment. And this can usually be achieved by proper biosecurity, vaccination, and flock health management program. So it is difficult and expensive to always maintain a high level of biosecurity, and those poultry vaccine and health management program are powerful tools in disease prevention and control. So this is the three important factors that for the prevention and control of the diseases. First, we focus on the environmental factors, which is involve a proper.
Excuse me, Professor Pija.
Yes.
Yes, please the restart from the first.
Or the slide not moving?
Yes, we are currently a little bit problem. We just to solve the problem, and please again from the start.
Oh, I need to start again?
Yeah.
Okay, sorry.
Yeah.
Okay, sure.
We missed the 2 or 3 slide.
Okay.
Thank you. From here. Yeah, okay, that's good.
Okay, thank you. Sorry. Okay, so as the indirect user of the HPC, so I'm going to share the application of the HPC in the fight against avian infectious diseases. Okay, so this is a topic overview. So I will talk about the poultry industry in Malaysia, the poultry diseases, prevention and control of the diseases, and how bioinformatics could transform the poultry health monitoring with the help of the HPC application, and also a bit on the research highlight and development that relate to the bioinformatics and also the HPC, and how also the bioinformatics together with HPC leverage the vaccine technology advancements, and conclusion, and also the way forward. Okay, so the poultry industry in Malaysia has grown significantly at about 5% annually, and has contributed about almost 90% of livestock production and 75% of the farm value of the livestock industry in the country. So basically the livestock is the second highest contributor to the value of gross output for the Malaysian agriculture sector, and it contribute about 18.7 billion, where the poultry itself contribute around 16.8 billion. So in Malaysia is self-sufficient for poultry meat and eggs, where Malaysia consumption is among the highest in the world, where about more than 50 kg per capita per year. So the industry is run commercially by large-scale integrators and also multinational corporation, and is an example of a success story in the livestock industry in the country, and perhaps acts a model of the livestock industry. The rapid growing of poultry industry, however, not free from many issues and challenges such as the high production cost and emerging and also the re-emerging diseases. To date, almost all non-major poultry diseases have been reported in Malaysia. So if you can see from this slide, so we have the pathogen that cause infection from virus. For example, we have the NDV, infectious bursal disease, the infectious bronchitis, as well as the bacteria, fungus, and parasite. For example, like Salmonella, Pastorella, and also the Mycoplasma. So the prevention and control of poultry diseases involve complex understanding of the interaction between the agent, host, and environment. And this can usually be achieved by the proper biosecurity, vaccination, and also flock health management program. It is difficult and expensive to always maintain a high level of biosecurity, and those poultry vaccine and health management program are powerful tool in disease prevention and control. So basically, these are three important factors in prevention and control of the disease. So from the environmental factors, we need to improve in terms of the proper biosecurity, cleaning and disinfection, and also good farm management practice. And the second one is the host or chicken factors, where we need to implement the effective vaccination and monitoring program, as well as chick quality and also good farm management practice, right? And then the third one is due to the agent factors, where the accurate diagnosis of the disease, detection or isolation and characterization of the agent need to do frequently, and effective routine virus surveillance and also monitoring program. Besides that, also need to have effective treatment, for example, like antibiotic and also drugs. Okay, so I focus more on the third part, which is the agent, where the whole process of accurate diagnosis is based on history, clinical sign, growth, circulation, and also the detection, isolation, characterization of the pathogen, right? The isolation and characterization of the pathogen is very important, as it deals with the laboratory technique, which focus more on the PCR and the sequencing, also sequence analysis, which is the gold standard to characterize or identify the pathogen. So basically, these services are handled or run by the government, university, and also the private diagnostic laboratory service. And my lab is also one of the laboratory that provide the services for diagnosis. Okay, so as you can see here, our Institute of Bioscience is one of the higher institution center of excellence. Starting from the April 2010, and then we focus more on the animal vaccine and therapeutics. Okay, so this is the ecosystem for our laboratory, where previously we focused more on the vaccine and diagnostic, which is we focus on the design and develop the vaccine, and also do some services on disease diagnosis. But now we are moving forward to the omics bioinformatics, where we deal with the big data, and then we do a genomic selection for poultry breeding and genomic surveillance for the disease diagnosis and also the prevention. And besides that, we also have the facility for the avian research and isolation, where we did the vaccine potency assay, vaccine efficacy study, and also the improvement of poultry breeding program. Okay, so how actually the bioinformatics could transform the poultry health monitoring indirectly using the HPC application. So traditionally, the bioinformatics is the science of storing, retrieving, and analysis the large amount of the biological information to solve the biological and also the biomedical problem. And this bioinformatics also is the interdisciplinary field that involve many different type of specialists, not actually limited to biologists, and but also involve the computer scientists and also the mathematician. So for now, the bioinformatics is actually combined the computational and statistical approach in order to analyze the biological data such as genome sequencing. So basically, this figure shows you the broad overview of the different type of data that fall within the scope of bioinformatics. So as I mentioned, before bioinformatics is used to describe only the science of storing and analyzing, but the term now is used more broadly, which is include the computational structural biology, chemical biology, and also the system biology, both data integration and also the data modeling of system. So beside that, the field of provide unlimited approaches for researchers to focus from only genes, genomic, only transcript, referring to the transcriptome, or also only protein, we're referring to the protein. So each level of investigation in answering different sets of questions. So this science field has potential to reveal the dynamic changes between the infectious agent, genetic material makeup, or protein profile in very short amount of time. So HPC is the revolutionizing the field of genomics by accelerating the speed of analysis and process large-scale sequencing data set. Hence, we had applied bioinformatics technology, which had adopted the proof of HPC system in the pipeline and also the software to solve the avian diseases issues and provide a suitable genomic surveillance protocol. So in our research, sequence genes were pre-processed and applied into an assay of bioinformatics pipeline, such as reference-based mapping, and also functional annotation and another more. So bioinformatics software often adopted the parallel and also the distributed computing system for an efficient, accurate, and also time-saving features during the analysis. Hence, in our genomic surveillance protocols, we use the whole genome sequencing, RNA sequencing, and also the shotgun metagenomic sequencing, which is incorporated pipeline with the HPC environment. So whole genome sequencing provide the most comprehensive of an individual genetic variation, which is include the genomic profile information, protein function annotation, also including the mutation, insertion, deletion, and also the structural variation. Okay, so this I caught one of the study that mentioned that by whole genome sequencing, we need the parallelization of memory power during genome read mapping and allows a significant complete of genome drugs. A rapidly growing field of science, it has shown great promise to help monitor, identify, and track poultry flock health. So when bioinformatics is associated with a program for monitoring poultry health, it can be used to detect early infection that may help may affect the productivity of the flock and thus allows the early development or preventive or corrective management practice. So data analysis allows for quick implementation for corrective management process. Okay, so this is another study by expert team in Poland and China where they took the bioinformatics as their approaches to retrieve quick and reliable genetic information of bacteria or viral strain that lead to the poultry infection. So both labs have performed the whole genome characterization to solve poultry disease outbreak in respective countries. And recently, Malaysian researchers has investigated around 11 IBD infectious bursal disease virus isolated from the commercial poultry farm. And surprisingly, 2 out of 11 isolate were identified as a variant strain which first reported in Malaysia, and highly similar to the novel Chinese variant. So the message I want to convey is that by decoding the genes, we can actually investigate the pathogen past, the origin, and the passage among family, and also the journey, and how the pathogen persists, how long it persists, I mean in the samples, okay, and also in the host. And also we can investigate the mutation and reconstruct a tree of the viral or bacterial descendant. And this all information will point the way to the vaccines, antibiotic, diagnostic kit, and also the monitoring program and public health strategy. So next, we move to the RNA sequencing. We did more study on the chicken transcriptome. So this use the distributed computing environment, which is allow functional annotation process of large transcript data to produce comprehensive information of the large transcript data. Okay. So in bioinformatics, the high throughput data is analyzed systematically, and results were generated according to the pipeline and software used. So the whole process will eventually lead to the, of course, the new finding and also the discovery. Okay, so this is some of the study that I have done. Okay, and others you also do a metagenomics profiling to observe any evidence of the antimicrobial resistant gene that released from the poultry or farm to the community, and assess the impact of the efficiency of the poultry waste management and also the wastewater treatment by study or analyze their alpha diversity microbial community profile in the phyla and genus, and also using some selective measure like Shannon and Simpson method. And we also perform the gene network of nodes and age of ARG to species, and also the species to species via the GEFI software using the span rank correlation value. So using the bioinformatics also, we able to leverage the technology vaccine advancements, where we study the bioinformatics of pathogen genome, profiling of host pathogen interaction, and also the pathogen genome editing. Right, so this is one of the research that we use the bioinformatics, how actually we modify the genome of the virus and then use that virus as a vaccine candidate to develop a novel attenuated vaccine. Okay, so as a conclusion, it is important to detect any genetic diversity and changes in genetic material of the virus and bacterial pathogen to solve the poultry infection outbreaks. The bioinformatics visually shine the light to save our poultry industry from becoming infectious poultry agencies, and indirectly fit the socio-economic balance. So the bioinformatics provides a great promise in monitoring, tracking, identifying, and protect our country commercialized poultry industry. So the exponential growth of HPC technology will enhance the genomic surveillance protocol and enable researchers around the globe to combat against the avian infectious disease holistically and also the efficiently. So however, we have some limitation and of course the strength of HPC bioinformatics relationship. So for the strength, of course, it will contribute to the time to workload ratio, and also the cost effective, and it also contribute to the time saving in producing comprehensive amount of data. But for the limitation, we requires an IT background personnel to assist in managing HPC environment, and also we are lacking a proper HPC facilities. Hence, that's why we open for collaboration, and then we can become a case study for HPC performance, something like that. So if people ask me how extreme HPC require in bioinformatics analysis, it's actually really depends on the data and choice of the tools used. First of all, see the latest boom in the data produced by these technologies. Of course, we need the high spec of the computing system. So, of course, the accessing any cloud computing service would be better. Other than that, it still depends on what on your downstream analysis. For example, if you use the de novo assembly, we expect higher spec, maybe Linux-based server, and also it's depend on the parallel amount data that you wish to analyze, and also on the application you choose. And because in bioinformatics we use a different platform like 454 or MySeq or from Illumina. And this machine generate different type of data, and then depend also what type of analysis that you want. For example, like whole genome assembly or transcript assembly. So then it will be need multiple core CPU, better GPU, and also huge RAM. For larger genome requirement, it can go up to half of terabyte, maybe, or maybe more. So if there are already available reference genome, and that we can have less RAM. Okay, and then however, for bioinformatics analysis in sequencing, the RAM is a very essential and very important. But of course, not forgot to the CPU, because if you want to run simultaneously, so we need more core. But for now, I think for us, about 16 core is enough for the individual, use for that one. And then for bioinformatics also, if for GPU, I think if people that works with the molecular dynamic simulation and plotting work, so yeah, they need more GPU. But for us, we'll need less GPU. But of course, now there are more integrated between the GPU, also CPU, and also the RAM. So with that, thank you.
Thank you so much for your presentation. And anybody question? Okay.
Thank you for a great presentation, Professor.
Okay.
Are you using any of the Illumina tools like Space, using their FPGA to do your alignment and analysis yet?
Sorry, again?
Are you using any of the Illumina sequencer tools and their environment like Space, which uses FPGAs to actually do the alignment and variant calling?
Okay, actually, in our institute, we have the Illumina platform, but the small one is the MySeq. So we tend to do the sequence, the small genome, like more likely virus and bacteria. So yeah, we use that one. But if you want to go for the something like the metagenomics analysis, that we need to do the genomic surveillance from the farm, for example, and then deal with big data. Normally, we will outsource, because we are lacking the facility that need to assembly, because that one need to handle very big data. And then after that, we get the data, then we process internally. So normally we work like that. But if the small genome, we will process here.
Second question, how do you see artificial intelligence systems fitting into actually matching your pathogen profiles going forward, or identifying new pathogen strains?
Sorry, you mean the AI platform?
Yes. Do you have any plans to use AI going forward?
Yeah, actually, I'm thinking of that, because we're thinking to improve on the what we call the genomic surveillance program. So maybe we can implement with the AI. If we able to get selected few markers to detect, for example, like the real pathogen in the farm, because depends on the heat or something whatsoever. We have proposed some study like that, but unfortunately, I think we need a partner to do that. because we are purely on the biological data part. So we need help partner if we welcome for any collaboration for that one.
Thank you very much.
Okay, thank you.
Thank you again, Professor Fija. Yes, yeah, currently it's over 10 and this minutes. And the next speaker is Asif Han, is from remote. He stayed in the Qatar, Doha. So I think he got some problem to access to FUBA. So I ask again the session 2, and I want to add in his presentation, the next session. Thank you for coming the session 1, the APRP working group. Thank you.